Thank you, everybody. Thank you for being with us today. What I thought that I'd do today is to really share with you our conviction that in the next 75 years of this century, science will be fundamentally computational. I don't have to convince you about that, but as a company that is focused on life sciences and technology, this is what we are doing, this event today. Maybe the best way to really represent that is really to focus on data sciences rather than AI. And my topic today is really that over the last 25 years, we have seen major data gathering consortia with analytics or advanced analytics, but little AI. And yet, only in the last years, and yet we have made enormous progress. So imagine as we put everything that we have seen today on top of this data and new data that will generate, it will transform, as you have been hearing at an enormous speed, transforming science into a computational discipline mainly. And I thought that maybe I go back and again, this is my biased view, how all this started, and maybe I go back to the Human Genome Project was a step change in how did we do science. Step change. Of course, it was important because it gave us the 20,000 proteins, was a catalog of this, but it was most notable because it changed the way in which consortia of large groups at the beginning, 20 sites in the 90s and maybe 500 investigators, 1,000 investigators, actually gathered data, and it set a completely different trend of how do we do science. And of course, this was followed by the HapMap, the haplotypes, was followed by 1,000 genomes over the 12, 15 years to the pan-genome. And in this trajectory, we got, of course, all the human genes. We realized that junk DNA, some of you are from my generation, that's how we call the spaces in between protein coding genes, was actually super important because coding regulations. We identified 1 million single nucleotide polymorphisms that actually was the basis for GWAS and genome-wide association studies that discover, polygenic risk scores for cardiovascular disease, for schizophrenia, set up, fundamental knowledge about 50,000 transcripts, etc. So that was a fundamental base layer of data that keeps growing and provided the basis for genetic and genomic diversity worldwide. And of course, the impact was first, it was really notable in 2 areas. With that data that was generated, in some cases there was additional data, in other cases it was just computationally, basic analytics and computational approaches on those data, no AI. We have fundamental transformation of rare diseases first. In 2001, there were 1,200 known at the genome. Today, almost 5,000. Clearly identified Mendelian and non-Mendelian. And that changed rare diseases, remember? And some of our companies are fundamentally involved with this. Integrated DNA technologies is at the core of how medicines were read, how these mutations were read through sequencing and now how they're being treated. Also Aldevron is fundamental there. And it changed completely how we do it. From the time to diagnosis, the use of therapies that were not going to be efficacious anyway are not being used anymore. 25 rare disease drugs for Mendelian diseases approved, another 25 failed in this period of time, due to mainly safety considerations. Complete change in genetic diseases. Important that, also Mendelian recessive diseases change geographically and in families. For example, 20 years ago in Saudi Arabia was the place with highest incidence of rare Mendelian disorders. Today, due to screening, and almost every gene that was responsible for recessive Mendelian disorders in Saudi Arabia is characterized, and many of them prevented. Also, Mendelian traits that were like Tay-Sachs syndrome or thalassemia that were particularly problematic in areas like New York or Cyprus are now screening programs that are being generated. So impact on genetic disease is important, but also on genes that are Mendelian genes that are accelerators for common diseases. There are 59 that have been identified as Mendelian genes that actually are at 2 or 3% in the population. A recent paper in the UK showed that 2% of us carry one of 59 genes that are of high risk of lethality. And of course, we have different polymorphic variants. So completely different change in how do we understand genetic disease and human variation. The other area of application, of course, was cancer. The Human Cancer Genome Atlas, TCGA, has been a fundamental engine of data. Over 12 years, it evolved into many different ones. The Pan-Cancer Atlas, 2.5 petabytes, this is 10 to the 15, 2.5 petabytes of data that have enabled investigators going into that database and really understanding, and what you have here in two sites is cancers like germ cell cancers that are particularly susceptible to chemotherapy and they respond very well, understanding what is the genetic basis of this, to cancers like head and neck squamous cell carcinomas that are particularly bad, that even after surgery, chemotherapy, and radiation relapse within 2 years with bad outcome. And now we understand what are the pathways that are getting there. So by going to these enormous databases that we generated over 20 years, we have been able to advance fundamentally our knowledge of some of these diseases. And by the end of 2020, through a number of different expansions of the TCGA, we have gotten to a good catalog of cancers, almost 22,000 cancers from 33 different types, very well understood at a molecular level. The Pan-Cancer Genome was actually the first distributed cloud computing effort in genomics. This is 2020. And if you have to compare this with actually the 1998 Human Genome Project, there was, I don't know if you have seen this, there were volumes printed in paper with the sequence of chromosome 17. So we couldn't really access those data because it was like an encyclopedia, like the Britannica that my father loved. So now is distributed cloud computing, we access this data, it changes our ability to deal with all these situations. On the right-hand side, of course, you have our colleague Faisal Mahmoud presented, was not only enough to have genetic variation, but it was important to put it in the context of spatial distributions. And around 2015, 2016, some of these considerations started to be made. And he and his colleagues published a paper with 14 cancer types where suddenly you combine molecular transcriptomics with histology and a weakly supervised model that they developed at the time actually was able to predict outcomes, favorable or unfavorable, based on the molecular characteristics that were associated to histology. So fundamental transformations. Of course, this was just genes. What happened not 20 years ago, but 3.5 years ago, was that structure was fundamentally transformed. If you look at the history of biology until 2021, we were able to elucidate one at a time, 100,000 structures. And in 2021, between AlphaFold and Rosetta Fold in different ways, and of course that's why there were 3 Nobel Prizes this year in chemistry, 300,000 structures were in one soup characterized. Since then, there are approximately 250 million computer-generated structures. around 100,000 of them human. So the transformations of how do we model interactions, and of course there are differences in how the two approaches generate that, are fundamental, and I'm not going to get into those, but I think they are fundamental for drug discovery, etc. What you have at the right-hand side is papers that came out less than 1 or 2 months ago with AlphaFold 3 and Rosetta Fold of all atoms that allow not only protein structures, but protein structures and the tokenization of other biomolecules allow still a quite static, not dynamic interaction, but actually a completely different set of that. So fundamental transformation that enables now, this is the Genome Project 20 years ago. Imagine what we have done. This is just the beginning. If we start to learn from that. Not only that, but remember before we were talking about understanding human variation through sequencing. We know all of them, but we don't understand diversity. Another important milestone from my perspective, for our perspective, is that, alpha missense and other algorithms allow you to, with 90% accuracy, to predict if the genetic variation that you have is going to relate with a poor outcome or is neutral. Important also to generate hypotheses. Around 2015, we started to think about, as a community, that we have to put all this in the context of a spatial information. And spatial started to occur. And I put here 2 papers. One that deals with the mammalian brain, the first draft on mammalian brain on your left. We have 90 billion neurons. Our colleagues here map like only 4 million, but they did transcriptomics and fish. And they discover the beginnings of fundamental differences between dorsal and ventral lineage determination in the space. That together with the connectome, we're talking about digital twins, talk about a digital twin. This is not mammalian, it's a vertebrate, it's the fruit fly, but the Flywire consortium, another consortium, was able to map 50 million synaptic connections and generate a digital twin of the whole vision of a fruit fly, where they can model, etc. This is just the beginning. Of course, there are a billion cells being targeted at the moment for the next generation of these maps, and this will set the stage for our learning and our ability to do integrative neuroscience. I want to finish maybe with the right-hand side. Less than a month ago, the first drafts of this human cell atlas were published. This started in 2016 with 100 scientists. Now it's more than 3,000 scientists that have been able to map around 65 million cells. Remember, we have, 37 trillion cells in our body, so there's a long way to go. 1 billion is expected to be completed within 2 or 3 years. So this is a long-term program, but this is the Google Maps of health and disease. And that ability to map, to share, to build consortia, together what we have been reading today, will transform science in an accelerated way. And in fact, at Danaher, we are preparing, for all the reasons that you have been hearing today, a really different 75 years of science. Because the scientific method, as we knew it, certainly as we did in the 20th century, is going to be, or is being changed. We are going to move from from physical experimentation, that will continue to occur, as we heard today, to generate data. Experimental, physical experimentation that is hypothesis-based, I think with statistics or computer analytics, is going to be transformed by hypothesis question, by computation of the data that is being generated. Science in this century, because there are more autonomous agents, as we heard today, they increase in the last year by 100 times the types of experiments that do not involve a human. Because of the amount of data that we generate, 30% in healthcare of the 175 zettabytes, 10 to the 23 data that is being generated is in healthcare. Because of the global collaborations, science in the 21st century will be computational. And we at Danaher want to be part of that with you. That's what we are getting today together.